The olfactory system processes information about the identity, concentration, and quality of a wide range of airborne volatile chemical stimuli called odorants. Odorants interact with olfactory receptor neurons found in an epithelial sheet, the olfactory epithelium, that lines the interior of the nose. The axons arising from the receptor cells project to neurons in the olfactory bulb through openings in the cribriform plate, a sponge-like bone located at the top of the nasal cavity. The olfactory bulb is located at the base of the brain beneath the frontal lobes. An axon pathway known as the olfactory tract projects from the olfactory bulb to the accessory olfactory nuclei, the olfactory tubercle, the piriform and entorhinal cortex, as well as portions of the amygdala. The transduction of olfactory information occurs in the olfactory epithelium, the sheet of neurons and supporting cells that lines approximately half the nasal cavity surface. The remaining surface is lined by respiratory epithelium, similar to that in other airways, including the trachea and lungs. The olfactory epithelium includes several cell types. The most important of these are the olfactory receptor neurons, or ORNs. These bipolar cells give rise to small diameter, unmyelinated axons at their basal surface that transmit olfactory information centrally. At the apical surface, an olfactory receptor neuron gives rise to a single dendritic process that expands into a knob-like protrusion from which several microvilli, called olfactory cilia, extend into a thick layer of mucus. The mucus that lines the nasal cavity protects the exposed receptor neurons and supporting cells of the olfactory epithelium and controls the ionic milieu of the olfactory cilia. Mucus is produced by secretory specializations called Bowman's glands that are distributed throughout the olfactory epithelium. Two other cell classes, basal cells and supporting cells, are also present in the olfactory epithelium. This entire apparatus, mucus layer and epithelium with neural and supporting cells, is called the nasal mucosa. The entire population of olfactory neurons is periodically renewed. Olfactory receptor neurons are generated from dividing stem cells maintained among the basal cells of the olfactory epithelium. Olfactory receptor molecules are homologous to a large family of G protein-linked receptors. Odor transduction begins with odorant binding to a specific odorant receptor protein, which in turn activates the linked G protein. In its inactive state, the G protein is bound to GDP. Receptor activation causes the G protein to exchange its GDP for GTP. The activated G protein then activates the enzyme adenylyl cyclase 3, an olfactory-specific form of adenylate cyclase. The activated enzyme molecule converts a large number of ATP molecules into cyclic AMP. One target of cyclic AMP is a cyclic nucleotide-gated cation selective channel that, when open, permits the influx of sodium and calcium into the cilia, resulting in depolarization. The ensuing increase in intracellular calcium opens calcium-gated chloride ion channels. Both calcium influx and chloride efflux contribute to the cell's depolarizing response. This depolarization is conducted passively from the cilia to the axon hillock region of the olfactory receptor neuron, where action potentials are generated via voltage-regulated sodium channels and transmitted to the olfactory bulb. The receptor potential is reduced in magnitude when cyclic AMP is broken down by specific calcium-activated phosphodiesterase to reduce its concentration. At the same time, calcium complexes with calmodulin and binds to the channel, reducing its affinity for cyclic AMP. Finally, calcium is extruded through the calcium-sodium exchange pathway to return internal calcium concentration back to resting levels. Individual olfactory receptor neurons are sensitive to subsets of stimuli. Some olfactory receptor neurons exhibit marked selectivity to a single chemically defined odorant. whereas others are activated by a number of different odorant molecules. The responses shown here are from a neuron that responds to all three of the selected odorants. Another neuron responds to only one of the three odorants. Presumably, the particular response pattern of an individual neuron reflects the expression of a single odorant receptor gene within that neuron. A third neuron responds to two of the three stimuli. A large number of unique odorant receptors exist in most species. Most mammals, for example, have around 1,000 different odorant receptor genes. This receptor diversity is believed to establish sensitivity to the myriad odors that most animals can discriminate.